Okay, we were doing the life cycle of Lycopodium, which is commonly known as club moss. Okay, so this is the systematic position we have done in the classification of pteridophytes. So this is just a revision for you. Now, it is commonly called as club moss because it has club-shaped strobili. Okay, a club is like a baseball bat. Okay, and it has moss-like appearance. Okay, it prefers to live in moist, shady places. So, uh, the part of your house which is always wet, no, the drains, and which is shaded, if you go and see, you'll always see some mosses there, moss growing, no. So it's because they prefer to live in the moist and shady places, and those places should be rich in humus and organic matter. Okay, so it is represented by about 400 species. Most are tropical to subtropical in distribution. Well, obviously, no. If you think about it, most of the plants they belong in the tropics, because in the polar regions nothing can grow, nothing can survive. No, so it is obvious. But there are some species, okay. However, there are some species that are found in the Arctic and alpine regions. Okay, alpine means top of the mountain. No, this is many layers of the mountain. So we are finding it somewhere here in the alpine regions, and the Arctic. They obviously, you know, if this is the North Pole and this is the South Pole, here's the equator. Arctic is somewhere here. Okay, or here, the down, you know, below the South Pole. Okay, so in India we have 33 species of Lycopodium, and we find them in our own place. We will see them. We are in the eastern Himalayas, no, and the South Indian hills. So that's why I told you, in your house, in the shady places, you will find Lycopodium because it's endemic to our, I mean, it is present in our place. Okay, not endemic, it is present in the northeast, okay, in the eastern Himalayas. So in India, the common Lycopodium that we find is the Lycopodium clavatum. Okay, it is the most common species in India, and there are other species like Selago, Lycopodium selago, Hamiltoni, Phlegmaria, Serratum, Cernum, etc. Okay, the ones that are mostly common is Clavatum, Selago, Cernum, Phlegmaria, Serratum. Those are the most common ones. Now let's look at the habit. When we talk about the habit, it means when you look at it, how does it appear? Okay, when I look at the girl, if I ask about her habit, it means, is she short, is she fat, is she tall, is she thin? Okay, so when we look at the plant, we will see that the body is divisible into roots, stems, and leaves. Okay, means it is differentiated. And obviously they are herbaceous. No, they are not trees, they are not shrubs, right? They are not trees because only pine is like a pine. They are not big and tall. They are not shrubs because they don't have woody stem. So they are herbaceous. Okay. Now there are epiphytic lycopodiums. Means epiphytes mean those that grow on the top of tree trunks. Like for example, Lycopodium phlegmaria. Okay. One example for each is enough. You don't have to remember all these because it'll be too confusing. So, epiphytic algae, you will remember Lycopodium phlegmaria. Then, the terrestrial means found on the ground with the prostrate creeping stem, means on the ground. No, they grow like a snake, like grass. Those you can remember the clavatum. Okay. Then the terrestrial with erect stem, you can remember reflexum. Okay. Then you have some climbers, Lycopodium that are climbers. This is volubile. So I think it's enough for you to remember one example from each. This one you don't need. Okay. So this is the diagram. So I think you have drawn it in your practical. You can see these are the club shaped sporangia. Do you see them? So here also these are the sporangia are present at the tips. Now if you notice it is what kind of branching? It is dichotomous. Okay, it has two direction branching. Here also dichotomous branching, there also dichotomous branching. Now look at the leaves. No, don't they look like scales? See the leaves, they look like scales. This scan is not so good, but you have already drawn. So the leaves, they look like scales. No, they are this shape, long. Okay, oblong, oblongate. Then what else? Okay, now if we look here, we can see one root. No, so I think this is the creeping type. So you see these are the rhizoids. This is one long root. Okay, so you can have the root system or the rhizoid system. So I think the habit is clear from the image. Now let's look at the sporophyte. The sporophyte means we are talking about the main plant body. Okay, the diagram that I just showed you, when you look at it, just looking at it, the sporophyte, if you see a cat and a dog, you will know who's a cat, who's a dog from their shape, no, from the whiskers, from the tails. So that, just imagine that a cat and a dog, that is their sporophyte, which makes them different from the other. So even here, when we talk about the sporophyte, it means we are talking about the main plant body. Now the main plant body, we will call it as the sporophyte or the sporophytic. And why we term this one sporophytic and the another one will be gametophytic. So I've explained it a little bit when I told you about the alternation of generation, no? So as we go on, as we do the life cycle, you will understand that it lives in two kinds of plant bodies. In one generation, it will be sporophytic, and in the second generation, it will be gametophytic. How can I give an example? Oh yes, very good example is when you were a child, you were very different, no? Used to play, used to laugh, no? You play with toys. You were a toddler, right? You were a child or a toddler. No? When you were, let's say, as soon as you were born, 0 years to, let's say, 12 years. Let's say, then this one is your sporophyte, okay, when you are a child or toddler. Then from the year 13 onwards to 19, you were what? A teenager, right? So this, let's say for example, this is your gametophytic generation. Okay, so even as we have 3 generations actually: child, teen, and then adult, right? And then adult. We have 3 types of generation, but here in Lycopodium you have 2 types. So I think it's clear, ne? Okay, so if we look at the sporophyte, we will notice that they have a very slender branch stem. Okay, you saw the stem, right? When in the diagram, it was very slender, very slim, not just around this much. No, in the drawing also, it was very obvious that it was a slender, very slender branch stem. And then there were so many leaves. Okay, there's nothing new here, girls, only what I have shown you in the diagram just now. Okay, and what were the roots? They were dichotomously branched. Okay, very easy. The sporophyte exactly from the image. So now let us look at the root. If you cut the root, okay, if you cut the TS of the root, when we do in the practicals, we will observe the roots in this way. Okay, so the primary root or the first form root of the young sporophyte is ephemeral. Ephemeral means it will live just for a short time. Okay, just like the taproot, no, of the plants. Okay, just imagine that this is a rose flower. Oh, my rose looks bad. You had a taproot, no? Then you had those adventitious roots coming from the side. Remember, you have done in school. So this main root, no, this big taproot is your it used to possess, right, in these these plants, right, these other herbs like roses and all, no? But here in this Lycopodium, this root, no, it will die off, this main root. Okay, so it will die off, and then the adventitious roots they will possess. Okay, so in case of the erect species like Lycopodium selago, just one example is enough, the root arises from the basal part of the stem. Okay, but the species with the creeping stem, the roots all appear along its length. Okay, in the other image we had seen, no, remember that one? This is the selago, the root came from the base. This was for the erect, and then for the creeping one, the cernum, the root came here, here, there. It comes like that. Okay, these are the strobili. Okay, I'm just drawing roughly. So according to whether it's erect or prostrate, that's how the root will appear. Okay, so outside of the root we have seen, if it's a creeping, it will come all along, and if it's a erect one, it will come from the base. Now if we look at the anatomy, means if we cut the root, the TS, and we observe in the microscope, okay, then we will see that there are three parts. Okay, the outer epidermis, the middle cortex, and the inner stele. Okay, maybe we should go first to the image. Okay, just look here. Can I zoom it? Oh yes, very well, very good. So here these are the root hairs. Then there is one layer of epidermis. Okay, it continues till the lower. Then you have a thick walled outer cortex. See these cells? Okay, the cells which appear like this means it is very thick wall. Okay, this is very thick wall. It means the cytoplasm and nucleus are all inside. The wall is very thick. Okay, then what do you see next? You see then wait a minute. Okay, then you see the okay, we have an epidermis, we have an epidermis, then next is your outer cortex, then next is your inner cortex. Okay, so how do I show? First layer, this is one layer, then two layer, then three layer. Okay, so there is three layers. Okay, so after that you will find that there is these big ones, these are the metaxylem. And I want to tell you girls, when you draw, no, you have to draw exactly like this. Okay, you cannot just draw your own you don't have to draw the complete, you can draw at least half. You can draw till here maybe if you're running out of time, but you have to show these different types of cells. Okay, you can draw it large. How do I show? Okay, is there empty space somewhere? Okay, here, dot. Let me just start. Okay, suppose I drew like this. No, we found the I wish I could draw in the board. We found the epidermis. Then you draw the thick layered cortex. No, okay, maybe one more layer you will draw like this. Then then you will draw the middle inner cortex. No, like this. Yeah, I wish I had a board for you to draw. Computer to draw, it's so tough. Okay, I think if I trace, fine. If I trace here, you have drawn, no, these cells, you can draw them big so that you don't waste time. Then you drew these cells, no, these metaxylem with a gap there. Then you draw these smaller cells. Okay, don't give any space. Try not to give any space. Then you label them: epidermis, outer cortex, inner cortex, no, metaxylem, and then parenchyma. And then if you notice these, see, metaxylem and protoxylem, no? Let me just introduce you. Metaxylem, these are the older form cells, okay? But just like you were a small kid, no, small toddler, then you grow up to a bigger teen, then you grow up to a big adult, okay? Very fat, big adult, so fat, big arms, big legs, so fat, no? So here also, these metaxylem are the older xylem, and these protoxylem, these smaller ones, these are your young xylem, okay? The young ones, they will keep on growing in size and being pushed inside, okay? I think it's clear. So you can see these cells, all these smaller cells in between the, if this is the xylem, no, and this here, all these small cells, all these are the phloem, okay? So when you draw, you can use tricks, no? You draw very fast, but you can draw at half, you know? You can draw only this much, okay? This moon, half moon you can draw, because it'll spend time, no, trying to finish it all. If you are fast, then you can draw the whole, but it depends, no? Okay, so just by practicing the diagram, we have learned. Okay, we have learned that there is a root hair, then epidermis, then the cortex, then there's a metaxylem and protoxylem, and innermost we find the parenchyma. Innermost we find the. Okay, I'll take. Okay, so if we look at the TS of this the root, we find there's a single layer epidermis. Then we find a thick layer of outer cortex cell. Then you have another layer of inner cortex cells. Okay, then you had a xylem. Okay, then you have this in the middle, you have the phloem. Okay, so just by looking at the diagram, you can describe in your own words, no, if they are asked about the TS of the root. Okay, so let's look. The outer epidermis is made up of single layer cells that are thin walled, okay, and parenchymatous. So most of the cells are parenchymatous, okay. Only those thick layered cells, no, those are sclerenchymus cells. Now, when you were in school, you learned, right? Parenchyma, collenchyma, sclerenchyma. Parenchyma are the normal cells, okay, the normal cells of the plants and the normal cells of the animals, okay. Then you have the sclerenchyma, which is thick. The wall is very thick. Okay, this is your sclerenchyma. Then you have the collenchyma, which is colon shape. No, this one is found in the inner lining of our gut. No, here these oval shaped cells. But in plants, mostly you will find these two types: parenchyma cell and sclerenchyma cell. Okay. Okay. Okay, let's continue. We saw many root hairs, no, that came out from the epidermal cells. Okay. Now, what is so funny about Lycopodium is that the root hairs, they come in pairs. Okay, they need friends. They're so funny, no? They cannot go alone. They cannot grow alone. They need to come in pairs. So, next to the epidermis, we found the cortex. Remember the outermost thick cortex, and there was an innermost normal parenchymatous cortex, which is, okay, epidermis is the cortex. Next to the epidermis is the cortex, which is several layers in thickness. In the older roots, the cortex is differentiated into the outer and the inner cortex. The outer cortex usually consists of heavily sclerified cells. Okay, remember I told you sclerenchymus cells. So this term comes from their sclerified cells because they have thickenings, no, big thickenings. Okay, so why do they need such thick cells? Just like the bark, okay, if this is the pine tree, no, why is the trunk so thick? Because imagine how heavy this part is. So heavy. Okay, if this part is so heavy, no, if the legs are not strong, like me, I'm very fat. This is me, no. If my legs are not strong, then how they can support this the rest of my body? So that's why the cortex is sclerified or is thickened to provide mechanical support to the root. Okay. So the mechanical support is done, no, by the outer cortex. Then the inner cortex, they do not need to perform support. They are parenchymatous or the ordinary cell. Okay, ordinary plant cell, which has the nucleus, all those. Okay. Then if you look at the central part of the root, we will see that it has a protostele or a basic type of stele. Okay. The stele is monarch with one protoxylem group in young roots, but become diarch or triarch in older roots. Monarch means only one is there, okay, the xylem. If it's diarch, will mean two, okay, or three or four, okay. So the xylem is C or U shape with the protoxylem at the tip and tips of the C or the U shape. Remember, we had seen the xylem was like this. These small cells were your protoxylem, and the big ones here, these big ones, these are your metaxylem. Okay. So where did we find the phloem? We found them in between the arms of the xylem. Okay. Here we found it, the phloem. So it's easy enough, no? This is the image again. It's very easy. Once you have practiced the diagram, no, girls, you don't even have to go through the text. You know that. As long as you have practiced, so whenever you study, no, whether you're studying algae or bryophyte or pteridophyte or anything you're studying in the future, even if you're studying zoology, first what you do is you draw the diagram first. You draw first. You practice in your copy. You draw. Then you try to remember what comes after what. No, what comes after what. Then you don't even have to go through the text. No, you just go through once and you already know. Point 1 means outer epidermis is covered by root hairs. And point 2, you will say by yourself, there is a thick wall of outer cortex which is sclerified. Do it by yourself. You just look here. Followed by next point, you'll have the inner cortex of is parenchymatous, and it follows the thick wall cortex. Then by yourself you continue on saying, giving the points. Okay, it's very easy. Now let's look at the next. Now let's look at the next diagram. Okay, if we look at the vertical section of the leaf, okay, now here you are seeing one layered epidermis. Very good. Then the epidermis is covered by a cuticle. Means it's like your nails, no? Your nails, these are the cuticles, right? Your fingers, oh, so pretty this hand, no? See, the nails, okay, it's covering the cuticle. And then what did you have? You have these cells, all these cells, okay? These, all those cells which have dots, no? Those cells, you are calling them as mesophyll cells, okay, because they're found in the leaves. Then you look in here, you have your xylem and you have your phloem. Okay, I can't make out which is which. Okay, so I guess you cannot differentiate this one, just draw it as it is. Okay, let's go to the next. So you have remembered, no, you had epidermis covered by cuticle, then you have mesophyll cells, and then you had xylem and phloem. Okay, so let's see what is there in the text. The leaves are simple, eligulate. when we do Selaginella, no, you will find that every leaf, no, has something like this, known as a ligule. Okay, but in Lycopodium, the leaf is just like this. Okay, there is no ligule. Okay, so it is simple without ligule. It is sessile. Sessile means this is not there. Okay, this branch is not there. This branch is not there. Okay, means if this is the stem, directly the leaf will come out like this, come out like that, directly from the stem. Okay, so it is sessile and the leaves are small which unbranch mid-vein. Okay, so they are lanceolate in shape. Okay, so remember in the diagram, this is the leaf. It came out directly, you know, like scales. So this is lanceolate shape and it has just one single vein. Okay, there's no branching. Okay, like that, no. It is just one single mid vein like this. Okay, they're very simple leaves. They are narrow at the apex and broad at the base. So you have to draw like this, like that. Okay, the leaf, something like that. It's so difficult to use my finger to draw something like this. It's broad here, narrow, broad at the base and narrow here. Something like this. Yeah, something like that. And just one single mid vein. Okay. This word phyllotaxy means the arrangement of the leaves. Now, suppose it's a sunflower. No, you have your leaves like this. No, alternate. Like that. No, this is leaf 1, this is 2, this is 3, this is 4. Okay. Then what if you have leaf like this? This is okay, like this. This is leaf 1, this is 2, this is 3, this is 4. Means your leaves are opposite, are they not? Then, but in Lycopodium, the leaves are arranged spirally. Okay, like that, spiral. Means one leaf comes like this, like that. How to draw spiral? Like this, draw like that. From behind also they'll grow, okay, all around. This is the behind part, okay, I'm showing like this. All right, so the leaves, they are arranged this way, spiral. Okay, so phyllotaxy is the arrangement of leaves. So it is arranged like a helix. Okay, means like I showed, you know, like this, like a spiral, round, 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 but like this. Okay, round, round, round. It goes till behind, then again it'll go round, again it go round. Okay, but they are actually helical, means arranged round, but they appear to be opposite. Okay, or whorled, like a whirl. No, you know what's a whirl? Flower whirls, no, like that. No, you remember the whirls. Okay, they appear. Okay, in some forms the leaf bases are decurrent. Okay, here's the meaning for decurrent. Okay, I don't have to explain because it's already given there the meaning. So the leaves are all usually alike. Okay, they're like clones. Okay, and they have the same size and the same shape. So we'll call them as hemophily or isophily. No, homo means same. Homo or iso means same. So homophily, because all the leaves have the same shape and size. Now there's a type let's give the example of Complanatum. complementum and volutile, the leaves show dimorphism. Dimorphism means they have two shapes, one big and one small. All right. Mostly, I told you they have the same size and the same shape. But in these two species, that is complementum and volutile, you have two different kinds of leaves. Okay, small leaf and a large leaf. So we'll call them as heterophyly. Okay. The other one is homophyly. The other one is the heterophyly. Okay. So we know about the external looks of the leaf. Now let's look at the internal looks of the leaf, that is the anatomy. So the anatomy is so easy, no? We had a one layered epidermis which was covered by cuticle, okay, cutillarized. Now they are interrupted by stomata because obviously they are leaves, no? So let's look again. Okay, do you see this? You see this? Can you see these two, this one and this one? These are your stomata. These are the guard cells, okay? These are the guard cells or the opening of the stomata. Oh, I can't write, forget it. Okay. Okay, let's continue. Okay. So it is very simple. It has lots of cuticle, one layered epidermis, which has interrupted by stomata, and it has similar shapes of cells, which are known as the mesophyll cells, with large intercellular spaces in between them. Okay, remember in the diagram, the cells, they had large spaces. Why? Because they are the leaves. No, they have to take in your carbon dioxide and exhale oxygen. No, so that's why those spaces, they are not exactly empty. They are filled by gas. Okay, so the mesophyll cells have large intercellular spaces and chloroplastids are present. Okay, obviously plastid has to be present so that they can manufacture food. And remember, I told you that leaf has just one median vein. If you cut this median vein, okay, you will notice that it has a concentric vascular bundle, okay, with central xylem and surrounded by phloem. Okay, so remember the diagram we could not make out. It's just one is a xylem here, and then here all these other cells is the phloem. Okay, so this is the phloem, this is the xylem, and the rest are what cells? Mesophyll cells, no? Like that, the rest are mesophyll cells. Okay. Stomata are distributed on both surface of the leaf. Okay, that is, it is amphistomatic. But in some species, like complementum and volutile, they are confined only to the lower surface. Okay, and these complementum and volutile, remember, they had different arrangement of leaf. They had one big and one small. Not only did they have a different size of leaf, they are heterophyly, but they also have only lower surface stomata. Okay, we are done with the root and the leaf. Now we look at the stem. Okay, and again what we will do, girls, we will look first at the image. Okay, it's not very clear, but I think you can make out. Okay, so this is the outermost layer of single layer of epidermal cells. Okay, one layer of epidermal cells. Okay, then you have a 3-layer cortex. You have the outer cortex, this one, this, then you have the middle cortex, these larger ones, and then you have the inner cortex, these. Okay, so they all have different shapes. Okay, then inside the inner cortex, we find one layer of single cell, that is your pericycle. Okay, it's just a one layer thick. Okay, we found your pericycle. Then inside the pericycle we find, this is your, remember we did in stellar types, remember plectostele? This is the diagram of plectostele. Now these big ones are your metaxylem and these small ones are your protoxylem, and here surrounding it is the phloem. Okay, this is nothing but your plectostele. Okay, but something else is present here known as a leaf trace. Okay, leaf trace means from here, no, a leaf will come out. Okay, means this is the vascular bundle, no, which will go into the leaf, follow till the midvein. Okay, so I think you understand what it means, leaf trace. Okay, so okay, we saw the diagram, very easy. Now it has a star shape also, star shape of xylem, star shape stele also it has. Right, it has this shape of stele also it has. It has this type of stele also it has, and this type of stele also it has. So it's showing different arrangement of the stele. Okay, this one, the one we did is the plectostele. Then this star one is the, what is it, girls? Does anyone remember? Actinostele. This one I think I forgotten. This could be actinostele. I'm not sure. I've forgotten. Okay, we'll see in the text. It'll be there. Okay, now let's see in the text what they have written about the stem. So if you pick up a lycopodium, you will notice that the stem is quite weak. Okay, they are soft because they are lower plants. No, they are not like rose bushes or pine trees. No, they are weak. Okay, they are thin, that is slender, and they have rhizomes. Okay, so it is erect like Silago or creeping on the ground like Phlegmaria. That's not important. Okay, let's look at the anatomy. Okay, anatomy of the stem is that it is differentiated into epidermis, cortex, and stele. Okay, so everything inside the pericycle, then you have your xylem and you have your phloem. Everything this is known as a stele. Obviously, you have learned it, so you know. So the outermost layer is one uniseriate, means single layer of epidermal cells, and the walls of the epidermis are thick and cuticularized. Okay, because think about it, it's not a root that it has to absorb water. This is a stem. Stem is what? It's above ground, isn't it? And it has to be strong enough to support this what is this, strobilus, support the leaves and all, no? So that's why the epidermis has to be thick and it must have cuticle to protect it. Now the cortex is very variable in thickness and structure. In some species it becomes homogeneous, that is made up of one type of cells, and in others the cells undergo sclerification, means sclerenchymatous, no, thick layered. Okay, so in the diagram, I think it was the clavatum. You saw only epidermis, then there were some thick cells, no, the thicker cortex. So it has outer and inner portion of cortex. We have the outer cortex and the inner cortex. And you have the middle portion, middle cortex, which is not thick. I'll show you again. Okay, 1 minute. Let's see it again. Okay, let me zoom. Here, in clavatum, no girls, do you see? The outer cortex is sclerified, inner cortex also sclerified, but middle cortex is the ordinary parenchymatous cells. Okay, so you can describe from the image only. Okay, but in Cernum, the middle portion is sclerenchymatous, while the inner and outer are parenchymatous. So if we are talking about Cernum, it will be opposite. This middle one will be sclerified, will have thick cells, okay, and the other one will be, outer and inner will be normal parenchymatous cells. Okay, so the endodermis is the innermost layer of the cortex, showing characteristic Casparian bands. Okay, it's like a strip, Casparian strip. Okay, so how do you know that you have reached the inner part of the cortex? It's because there is the Casparian bands. So inner to the, after the endodermis, no? Okay, let me say again. You had the epidermis, then you have the thick, normal, and again thick cortex. Then you have endodermis, okay, endodermis, which has what? Casparian band, okay. Then after the endodermis, you will have one layer of what? Pericycle, okay. Is it clear now? I hope it's clear. So you have the endodermis, which is inside of the cortex, inner to the cortex, which shows Casparian band. Then after the endodermis, you will find a pericycle, which is 2 to 6 layer thick. Okay, so I have made a mistake. Endodermis is single layer, but pericycle has 2 to 6 layers. Okay. Okay, so very easy. Here from the diagram, you saw this single layer of epidermis, not epidermis, sorry, a single layer of endodermis. Then you have 2 to 6 cells of pericycle. Then you will have the innermost xylem and phloem. Now, just by looking, you know that the stele is protostelic because there's no pith. No, not like higher plants. You had a central pith. So this one directly, the innermost part is the xylem and phloem. So it means it's a protostele. But as I showed you, the vascular tissues, that is xylem and phloem, they show different arrangement in different species of Lycopodium. So they are showing like a star, actinostele. In the case of Serratum, Silago, Phlegmaria, okay, you remember only one. Okay, like a star, you remember only Silago. So here the actinostele, the xylem is star shape. Blah blah blah. I don't need to repeat, no girls, because we have done types of stele. Then you have plectostele, like the example we did, no? Like I showed you just now in, you remember, clavatum. Then you have mixed protostele in, you can remember, lateral. Okay, so then you have the different kind of cylinder in this. Okay, so this one you haven't done, no? Stellate form is when the xylem is broken up into irregular pieces. Okay, so no one has any idea which is, see, like here, no? This one, girls. This is the stellate type where xylem is broken up into different pieces. There's no particular shape. Like here, we know it's a star shape. Now, this also, it's going to become a star only. This one is a parallel plates. But here, there's no shape. Now, so this is your stellate type. All right. Example for stellate is lucidulum. Okay. Okay, so we are done with the, the sporophyte part. Okay, the now let's look at the reproduction part.